This video will explore the urea cycle, presented by Medicos Online. Urea biosynthesis happens in 4 stages: transamination, oxidative deamination of glutamate, ammonia transport, and the reactions of the urea cycle itself. The diagram illustrates these steps leading to urea formation. Cellular proteins are continuously degraded and synthesized in all life forms. Humans turn over 1 to 2% of their protein daily. The liberated amino acids are mostly reutilized, but the excess nitrogen is converted into urea. This diagram provides a broad overview of how amino acids, derived from dietary or intracellular protein, feed into the urea cycle and the citric acid cycle to produce energy and excrete nitrogen as urea. The urea cycle, also known as the Krebs-Henseleit or ornithine cycle, was the first metabolic pathway to be elucidated. Urea is synthesized in the liver and transported to the kidneys for excretion. The 2 nitrogen atoms in urea come from ammonia and the alpha amino group of aspartic acid. Urea makes up 80 to 90% of the nitrogen-containing components of urine. This image shows the core steps of the urea cycle. Ammonia and carbon dioxide form carbamoyl phosphate, which then reacts with ornithine to begin the cycle, eventually yielding urea and regenerating ornithine. Urea synthesis is a 5-step cyclic process involving 5 distinct enzymes. The first 2 enzymes are in the mitochondria, and the remaining 3 are located in the cytosol. This visual illustrates the compartmentation of the urea cycle, showing the mitochondrial reactions and the cytosolic reactions, with the 5 steps clearly numbered. Step 1 is the formation of carbamoyl phosphate. Carbamoyl phosphate synthase I, a mitochondrial enzyme, catalyzes the condensation of ammonium ions and carbon dioxide. This irreversible and rate-limiting step consumes 2 ATP molecules. Carbamoyl phosphate synthase 2, or CPS2, is involved in pyrimidine synthesis and is located in the cytosol. It uses glutamine as the amino group donor and does not require N-acetylglutamate for its activity, unlike CPSI. Step 2 is the formation of citrulline. Citrulline is synthesized from carbamoyl phosphate and ornithine by the enzyme ornithine transcarbamylase. Citrulline is then transported to the cytosol by a transporter system. Step 3 is the formation of arginosuccinate. Citrulline condenses with aspartate, incorporating the 2nd amino group of urea. This reaction, catalyzed by arginosuccinate synthetase, requires ATP, which is cleaved to AMP and PPI. Step 4 is the formation of arginine, or the cleavage of arginosuccinate. Argininosuccinase cleaves arginosuccinate into arginine and fumarate. Fumarate connects to the TCA cycle, or gluconeogenesis, where it is converted to oxaloacetate and transaminated to regenerate aspartate. Step 5 is the formation of urea. Arginase, the 5th and final enzyme, cleaves arginine to produce urea and ornithine. Ornithine is regenerated and re-enters the mitochondria for reuse in the cycle. Arginase is activated by CO2+ and Mn2 and is primarily found in the liver. The overall reaction summarizes the energetics of the urea cycle. A net total of 4 high-energy phosphate bonds are utilized per cycle. 2 ATPs for carbamoyl phosphate synthesis, and 1 ATP converted to AMP and PPI to produce arginosuccinate, which equals 2 high-energy bonds. The regulation of the urea cycle relies on carbamoyl phosphate synthase I, or CPSI, which is the rate-limiting enzyme. CPSI is allosterically activated by N-acetylglutamate, or NAG. The rate of urea synthesis in the liver correlates with the concentration of NAG. This diagram illustrates the formation and degradation of N-acetylglutamate, the allosteric activator of CPSI. N-acetylglutamate synthase uses glutamate and acetyl-CoA to form NAG, a reaction stimulated by arginine. NAG is then degraded back to acetate and glutamate by hydrolase. The urea cycle and the citric acid cycle interact via the aspartate argininosuccinate shunt. Fumarate, produced in the urea cycle, enters the citric acid cycle to become oxaloacetate, which then regenerates aspartate, linking the 2 cycles. The main function of the urea cycle is to remove toxic ammonia from the blood as urea. Defects in the cycle's metabolism can lead to hyperammonemia or ammonia intoxication. Inherited disorders of the urea cycle are characterized by defects in specific enzymes, leading to the accumulation of certain products. For example, hyperammoneamia1 results from a defect in carbamoyl phosphate synthetase 1, leading to ammonia accumulation. Ammonia toxicity occurs when increased ammonia levels in the brain lead to glutamate formation. This causes more utilization of alpha-ketoglutarate, a key intermediate in the TCA cycle. The resulting decreased alpha-ketoglutarate impairs the TCA cycle and reduces ATP production. Thank you for watching this video on the urea cycle.